I want to officially welcome everyone to CureAccelerator Live for Rare Diseases. My name is Barbara Goodman. I'm the President and COO of Cures Within Reach, and I'm joined today by my colleague Claire Thibodeau, who's our Director of Scientific Affairs. Thank you so much for joining us for this event. What you're going to hear today is a Cures Within Reach event that we call CureAccelerator Live, and you, the attendees, are going to help us select the winner. Before we get started, I want to share some brief housekeeping items for all attendees. First, all the attendees have been muted by the moderator to reduce any interruptions. We do encourage you to use the chat function in the Zoom window for any questions that you have through the session, and the moderator will help us share them. To open the chat, just click on the chat button located on the meeting controls at the bottom of your Zoom screen near the mute icon. And do note, please, that we will be having a Q&A session, but due to time constraints, all the questions will be led by our expert panelists. And we do recommend that for all the rest of the attendees, we recommend that you visit the poster session immediately following the pitch to ask your questions directly to our finalists. If you do have any technical difficulties or questions about the program as a whole, please visit the help desk in the main lobby where you can chat with the tech team and you can chat with the Global Genes staff members as well. Finally, again, we really encourage you to go to the chat sessions after and ask your questions to our finalists prior to making a decision. This event is your time to learn and find the answers on clinical repurposing trials for rare disease patients. And with that, I want to formally welcome you to the second annual CureAccelerator Live for Rare Diseases. We are thrilled to return as a partner to the Rare Drug Development Symposium led by Global Genes and University of Penn Orphan Disease Center. Briefly, Cures Within Reach, our mission is to improve patients' quality and length of life by leveraging medical repurposing. Our goal is patient-focused. We're driving more treatments to more patients more quickly. As a not-for-profit, our funding comes from individuals, foundations, industry, and other partners. As I said earlier, we are patient-focused. We support projects that both have commercial value as well as those projects that have philanthropic value that are making an impact on patients that may not have the commercial side. We are disease agnostic. As you can see from this slide, we have funded projects in roughly 50 different diseases. We are geographic agnostic. We've funded projects not only across the United States, but in Europe as well. And specifically in rare diseases, we have 20 ongoing funded projects right now. 40% of those are in rare diseases. So rare diseases, as many of you know, is a place that repurposing is a very easy, quick way to find treatments faster for patients. The other thing about us is our ideal time to fund these projects is that proof of concept pivotal trial that leads to follow-on funding. And as you can see on this slide, $6 million we've invested in these trials that has led to $49 million invested in our researchers and follow-on funding, catalyzing our funding further. We're very, very grateful to Goldman Philanthropic Partners, who's supporting the winning grant today, as well as support from our industry partners, Horizon Therapeutics, as well as Recordati Rare Diseases. We're grateful to Judy Hirsch Foundation, another philanthropic partner of ours. And again, we're very grateful for Global Genes and for the Orphan Disease Center of UPenn and this event for having us back again this year. A couple quick things to keep in mind about our program today. We have pre-recorded 7-minute pitches from each of our 5 finalists. You'll then have a Q&A session, 2 to 3 minutes of Q&A from our expert panels. Like I said earlier, that's when, after all that is done, voting will open, and then we encourage all the attendees to go into the chat room, the private chat rooms, and ask questions of those finalists as well. Voting will be open for 45 minutes this afternoon, and we'll be announcing the winner tomorrow morning at 11:20 Eastern time. With that, I'm going to turn it over to my colleague, Claire Thibodeau.
Thank you, Barbara. As Barbara mentioned, we have 5 outstanding finalists with us today. And not only do we have those finalists, we also have 5 incredible expert panelists who will be joining us, or who are joining us, to help with the Q&A session, as we mentioned. Their questions will help us understand more about the projects, provide a little context, and really help us understand how these might be impactful for patients. So now I'd just like to briefly thank Dennis Costello, who's joining us from CML Advocate Networks, and I might also say from Spain, so we appreciate that as well. Maureen Hart from Global Genes, who many of you have probably already seen today. She's having a very busy day, but is still making time to be an expert panelist. Jeff Sherman from Horizon Therapeutics, one of our sponsors, as well as Brian Voss from Recordati Rare Diseases, who is also one of our sponsors. And last but certainly not least, Scott Weir, who is with the University of Kansas Cancer Center. So thank you all to the expert panelists, and we look forward to hearing from you in a little bit. Now, one of the most exciting things I think about is a great feature of CureAccelerator Live is that every person who's attending the event today has the ability to vote for the winning project. So you all have a pretty big role to play in the decision. So to help you with that, we are providing some voting considerations for you to keep in mind as you listen to the pitches, as you chat with the finalists in the poster session after, to help you make that voting decision. First of all, the patient impact of this potential project. How will this project improve patient lives? The probability of success for the research to get the data that it's hoping to. How quickly will this potential treatment be able to reach patients and start making that patient impact? An assessment of the entire research team, not just the presenters who are with us today, but the others who will be helping them during the course of their work. And then finally, the overall presentation. So please weigh each of those considerations equally when you make your voting decision. So again, we have 5 incredible finalists who are representing outstanding research institutions from across the country. They come from California, from Michigan, from Pennsylvania, all joining us here today. And each one of these finalists are passionate about what they're doing, about helping patients, and they have these really interesting and innovative repurposing research projects. We can't wait to share them with you. So let's not wait, let's get to it. So we have each presentation pre-recorded, as Barbara said, that will be given in a random order, followed by Q&A, and then more presentations after that. So to get us started, we're going to start with Dr. Susanna Park, who's joining us from University of California, Davis. So if we can get her video going, we can get started. Thank you all.
Although this is a genetic condition, in a majority of cases, there is no known family history. To date, the only treatment we can offer our patients is nutritional supplementation with vitamin A palmitate, which has a very modest effect on disease progression and does not stop or reverse vision loss. In addition, there are potential serious side effects. Therefore, research is being done using gene therapy or stem cell therapy for retinal regeneration. In fact, the first gene therapy approved for by the FDA was that for a very rare form of retinitis pigmentosa associated with a specific gene. Unfortunately, there are over 100 different genes associated with retinitis pigmentosa and many more yet to be identified. Regarding stem cells, various companies have developed embryonic or fetal stem cell lines for retinal regeneration, but this area of research is early in development. We believe we can use CD34 positive stem cells in our own bone marrow for retinal regeneration. These cells have been used for decades for bone marrow transplantation to treat leukemia or lymphoma. Therefore, there are established protocols for stem cell isolation, and the cells have been well characterized. More recently, CD34 cells have been found to be natural repair cells in our body, and clinical trials have been conducted in patients with heart disease, showing that local infusion of these stem cells is well tolerated with potential advantages. We aim to harvest these repair CD34 cells from bone marrow and inject them directly into the eye, as shown here, in order to maximize the repair potential of these cells on the retina. The advantage of using CD34 cells is that you can use your own stem cells, which is safer than using embryonic or fetal stem cells. In addition, based on the known mechanism of action of these stem cells, the cells can be used for broad clinical applications. The treatment may not be limited to retinitis pigmentosa. For a new treatment to become standard of care, years of clinical research are conducted for various phases to obtain enough safety and efficacy information for the Food and Drug Administration. However, by using an established treatment like the CD34 cells for a new indication, that is retinitis pigmentosa, we can dramatically shorten this timeline. We have already conducted extensive animal research showing that CD34 cells can be safely injected into an eye with retinal disease, and the effect was protection and regeneration of the damaged retina. In addition, we obtained an investigational new drug clearance from the Food and Drug Administration and completed a phase 1 study showing that it is feasible to inject your own stem cells into eyes with retinal degeneration without any major safety issues. Currently, we received funding from the National Eye Institute and is conducting a larger phase 1/2 study to obtain more safety and efficacy information regarding the use of your own stem cells in eyes with vision loss from retinal vein occlusion, which is a very common cause of vision loss. In addition, we have started a larger phase 1/2 study to evaluate the stem cell therapy for retinitis pigmentosa. For the proposed study for retinitis pigmentosa, we aim to enroll 8 participants, and we have already enrolled 3. All 3 tolerated the treatment well and noted some visual gain. For the 5 additional participants needed to complete the study, we need $60,000, the budget which can be met with the help of CureAccelerator. This interdepartmental research at University of California, Davis involves our study hematologist, Dr. Betty, who performs the bone marrow aspiration, our good manufacturing practice laboratory director, Dr. Bauer, who performs the stem cell isolation, the director of stem cells at UC Davis, Dr. Jan Nota, who gives scientific advice, and myself. I am the study ophthalmologist and the principal investigator conducting the injection of stem cells in examining our patients. The goal is to complete the proposed phase 1/2 study, and if the results show safety and efficacy of CD34 cell therapy in patients with retinitis pigmentosa, we can validate the off-label use of this cell therapy such that the treatment can be available to our patients within 2 years. The ultimate goal is to improve the vision and quality of life for millions of people with retinitis pigmentosa or other degenerative retinal conditions. Thank you.
We are back live, and Susanna, thank you so much for that recorded pitch. I bet everyone is glad that these pitches are recorded and not live right now. It's a little less stress. So now we'd like to have the expert panelists start with the questions. Maureen, why don't you kick us off with the first question to Susanna?
Yes, thank you so much. That was a wonderful presentation. And I just have to say, just to start, I'm terribly impressed with all of the finalists. So I want to put that out right from the very beginning. So Dr. Park, I was really interested in this and you know, hearing a little bit more in the presentation that you just gave about the safety profile. And I was just wondering if you could just expand a little bit on some of the side effects that you may have observed in the animal trials and how those balance both against the improved outcomes that you see in the vision in the animal models, as well as the current therapy with vitamin A treatment.
So that's a very good question. Before we could get an investigational new drug clearance from the FDA, we had to perform extensive animal studies to show that injecting CD34 cells into eyes will not create tumors or abnormal cells growth inside the eye. And we have done that using not-scared mice with retinal vascular disease. And we followed the animals up to 6 months, showing that there's no abnormal proliferation of cells inside the eye or any other parts of the body. And we could follow these cells because they were human cells injected into mice. And the FDA was happy with our safety information and cleared the IND. So far, we've enrolled a total of 9 patients in the NIH study. We have possibly 3 additional patients because it's a randomized prospective study. And we haven't had any abnormal adverse effects inside the eye. In fact, even a day after the injection, except for a little bruise on the surface of the eye where the injection was performed, we don't see any changes inside the eye, inflammation, you know, pain. Those are things we're looking for. So as we go forward, we are looking for signs of abnormal cellular proliferation, infection. You know, if the cells are not isolated in a pure manner and you get contaminants, you can cause infections in the eye. That's another thing we're looking for. And so far, it's been a very good safety profile, much better than other stem cells that people have tried, where you can get inflammation in the eye using allogeneic fetal or embryonic stem cells. You can get abnormal cellular proliferation, and that's been observed using cultured cells, including embryonic stem cells that have been developed to partially differentiated into retinal cells. The nice thing about CD34 cells are that they're not actively dividing cells. We don't culture them in tissue culture before injecting. We get them directly from the bone marrow and within hours inject them into the eye. And we have an extensive testing done before the release of the cellular product for patient injection. The FDA set guidelines, so we make sure that the cellular product is clean and pure of bacterial contaminants. We make sure that the cells are good quality and quantity, and before they're actually released. And even after release, they continue to culture the wash solution to make sure there wasn't an occult infection that occurred during the isolation process. It's important to know that as we published our phase 1 study, people who are not in research have used that data to convince patients to participate in fee-for-service treatments of quote stem cell injection inside the eye. And these are not regulated studies. We don't know exactly what they're injecting based on anecdotal reports. They draw fat cells, sometimes bone marrow cells. We don't know if they're characterized. They inject them into the eye, sometimes by a non-physician. And there are reports of people having serious adverse events where the retina is totally damaged and the patient loses what remaining vision they have. And so I think it emphasizes the importance that this is still investigational, but so far the treatments, the cellular product that we've obtained from the good manufacturing laboratory sterile environment have been all very good, and they've been all well characterized. So if we do complete the study and can show that it's safe and effective, we will of course emphasize that you can't just go to any center. You have to go to a center where the cells are going to be characterized before they're injected.
Great. Thank you very much for that. And you know, I should note that because of time constraints, we have 3 to 4 minutes for this Q&A session. And, you know, it's very important to get, to be clear on that safety data, I think, especially with stem cells. Unfortunately, that's the only time we have for questions. We will now be moving on to our next pre-recorded. So thank you very much, Susanna and Maureen, for that, that really insightful question. Next up, we have Alex Seif from the Children's Hospital of Philadelphia. If we can get her video going, we will hear from Alex.
Bone marrow transplant specialist at the Children's Hospital of Philadelphia. We're going to talk about how we will be repurposing ultrasensitive testing and targeted immune therapy to prevent relapse in children who are undergoing bone marrow transplants for leukemia. My very first patient when I was an oncology fellow was an infant with leukemia. Like many infants, he ended up relapsing almost immediately, and his leukemia wasn't responding to any of the chemotherapy we tried. We sat his family down and prepared them for the worst, and around that time he developed a severe bacterial infection. Now, fortunately, he recovered completely from the bacterial infection, but surprisingly to me, at that time we discovered that his leukemia was in a spontaneous, complete remission. And this was an important lesson for me in how powerful the immune system can be in treating chemo-resistant leukemia. We have known for many years that leukemia that sticks around after chemotherapy needs a different approach. And traditionally what we've used is an immune therapy delivered as a bone marrow transplant. Bone marrow transplant is an intensive treatment that's reserved for children whose leukemia can't be cured by chemotherapy alone. It's our current standard of care and the best we have, but even still, about 50% of children will go on to relapse. And relapse after transplant has a dismal survival of only 10%. By the time a child has had a complete relapse, there really isn't that much we can do. However, we do know that if we find a small amount of leukemia quite early and act immediately with ways to stimulate the donor immune cells to kill that leukemia, in some children we are able to prevent a relapse. So that's the focus of this talk, and we think that preventing relapses will save children's lives. ALL, or acute lymphoblastic leukemia, is a rare diagnosis. It affects about 3,000 children a year in the United States. Incidence rises every year, and we don't really understand why. Most children do really well, but for the 15% of kids who do go on to relapse, most of them need bone marrow transplants, and only half of them survive. So we really need better treatments for relapse ALL. An exciting new therapy that has been in the news a lot is a chimeric antigen receptor, or CAR-modified T cell. This is a game changer for many children, but it's not an option for everybody. And the cost is prohibitive in some patients, up to $1 million to manufacture and deliver this treatment. And then about half of the children go on to relapse after this therapy, which underscores the need to develop better relapse prevention approaches for after transplant. Our first repurposing goal is to take a brand new method to detect leukemia at lower levels than ever before, called next-gen sequencing, MRD, which stands for measurable residual disease. When a pathologist looks under the microscope, they can find about 1 in 500 or maybe 1,000 cells. MRD evaluates millions of cells at a time and can find very, very low levels of disease. Our current standard type of MRD finds about 1 in 100,000 cells, but this new next-gen sequencing MRD can find 1 in a million cells. Now, we know that children who are coming into transplant with detectable next-gen sequencing MRD are at a significantly higher risk of experiencing relapse after transplant. What we don't know is the best way to use this test after transplant and whether intervening makes a difference. Our second repurposing goal is to find novel immune therapies that can be used to get rid of MRD more effectively. Our current approach when we find MRD relies on ways to stimulate the donor bone marrow cells, immune cells, to find and kill leukemia. If kids are on immune suppression to help protect them from donor cells, we can take that immune suppression away, or we can give more donor cells to try and stimulate an immune response. However, neither of these methods are very specific for eliminating leukemia cells, and both of them can have really significant severe side effects. And then if the disease levels are high enough, neither of these treatments really work very well. But now there are targeted therapies available that are approved in other diseases that could be more effective at eliminating MRD after transplant. First, there's a drug called daratumumab, which is approved in an adult cancer called multiple myeloma. This type of cancer actually shares some features with ALL, and we expect that this drug may have an effect in ALL as well. Blinatumomab is a second agent that is already approved in relapsed ALL, but has not yet been used in a post-transplant relapse prevention setting. Our study is the PREEMPT ALL study. This is an initial pilot feasibility study with a goal of sending off this NGS MRD testing and getting children into an individualized targeted therapy within 14 days of sending the test. Treatment for each child will be individualized based on their specific leukemia's markers. We have a 3-arm study with 10 children per arm, a daratumumab arm, a blinatumomab arm, and a standard therapy arm if a child has a leukemia that isn't expected to respond to the other 2 drugs. There's so little that's known about using this type of testing and these treatments after transplant that we think the results of this pilot study will be a standalone publication. But really our goal is for this study to support development of a safety and efficacy study. And we currently have a proposal for a 3-year phase 1/2 safety efficacy study under review at the Gateway Foundation. At CHOP, we have a clinical research infrastructure through the Center for Childhood Cancer Research that can support our sample storage, processing, regulatory needs. And we also have one of the largest bone marrow transplant centers in the country and have every expectation that we'll be able to enroll on this study quite quickly. Our study budget accounts for a clinical research assistant to help run the trial, patient costs to offset travel and any clinical costs that are not covered by insurance, and a modest supply budget to collect and store biospecimens for future studies. Our team is comprised of an expert in relapse prevention and immune therapy, a clinical trial expert who is internationally renowned in ALL and pioneered daratumumab in mouse models of ALL. We have an experienced leukemia and bone marrow transplant nurse practitioner who will manage the clinical care and therapy delivery for the study. Of course, our protocol support office will help us run the trial. And we have a hematopathology coordinator who will be key in making sure that we streamline the testing process so that we're able to meet our feasibility endpoint. We know that effective relapse prevention will save lives and reduce the need for costly treatments like CAR T cells. We already know that far less sensitive leukemia detection and crude interventions can be effective for some children, and we expect that these more precise measurements and treatments will be even more effective at preventing relapse. The tests and treatments are already clinically available and can translate immediately into widespread clinical use, and we have a large multidisciplinary team that is ready to take this study forward. Thank you very much for your time and your consideration.
All right, thank you very much, Alex, for that, that great presentation. Scott, do you have a question for Alex?
Yeah, sure, Alex. Great presentation, thank you.
Thank you.
There appears to be little safety experience with daratumumab in children. And I guess, could you describe to everyone how you will monitor safety in the Stratum 1 treatment arm, and then whether your safety monitoring plan has been reviewed with FDA?
Absolutely. So there
Anecdotally, we have used daratumumab in children at the Children's Hospital of Philadelphia, both in the non-transplant and post-transplant setting. Certainly we need to monitor for cytopenias and other toxicities, although that is the main side effect that the adult trials have seen and that we have seen. The daratumumab is being evaluated to roll out in pediatric T-cell ALL studies based on Dave Teachy's work, who's one of my co-investigators. That's a great question. We have not yet run our safety monitoring by the FDA. However, based on our experience in a small number of patients, basically the equivalent to a pilot study's worth of patients, and the very limited side effects that we've seen, we expect that we won't have any issues.
Great, thank you.
Thanks, Alex. Let's see, Maureen, do you have a quick question for Alex?
Well, I just have one quick question, because one of the things that I was really excited about this is just to kind of confirm, this is really looking at sort of multiple levels of repurposing, because you're looking at not just trying to repurpose to think about prevention, but also thinking about repurposing a separate therapy or a separate drug in order to see if it will help with prevention as well as a particular biomarker for relapse. Is that correct?
That's correct. Yeah, so our goal here is to really combine two approaches to come in at the earliest possible time point. I will also add to Scott's point, we know that with many of these immunotherapeutic agents, coming in at a lower disease burden level actually attenuates many of the side effects that we see. CD38, which is the marker that daratumumab targets, is expressed on many different cells, but at least in our experience in the targeting CD19, we have seen minimal toxicity, both of blinatumomab and CAR T cells, when we come in at a low disease burden. So coming in at this truly, almost undetectable disease burden, we are hoping will address safety issues, will bring novel targeted therapies to these kids, and coming in as early as possible, we hope will allow for better successes than what we've seen when we've intervened at higher disease levels.
Excellent. Thank you very much, Alex. Appreciate it. And thank you for your presentation.
Thank you.
So now we'll move on to our third finalist. We have Angela Fleischman from the University of California, Irvine, sharing her proposal.
Fleischman, a physician researcher from the University of California, Irvine, and I'd like to share with you some exciting recent findings from my lab and our plans to develop this into a clinical trial testing the widely available antioxidant nutraceutical N-acetylcysteine in a very rare blood cancer called myeloproliferative neoplasms. I've dedicated my career to finding new treatments for patients with myeloproliferative neoplasms, or MPN for short. Although MPN is rare, affecting only about 1 in 1,000 people, it has many similarities to a recently identified precancerous state that's seen in about 25% of people over the age of 65. Treatments that we identify for MPN could possibly then be expanded to this wider population of precancerous patients to prevent blood cancers. MPN happens when a blood stem cell develops a mutation which leads to uncontrolled protection of blood cells. There are 3 major clinical issues in MPN. 1, thrombosis or blood clots, which in some cases can be life-threatening. 2, debilitating symptoms such as itching and fatigue, which can negatively affect patients' quality of life. And 3rd, MPN patients can develop an acute leukemia. And when they do, this has a very dismal prognosis. There are real trade-offs for our current treatments of MPN. We do have some treatments that prevent thrombosis, but unfortunately, some of these can actually worsen symptoms And even more alarmingly, it's possible that long-term use of some of these medications can actually increase one's risk of leukemia. In the symptom realm, we have JAK inhibitors, which work pretty well, however, not without a very high price tag, on the order of about $8,000 a month. Moreover, JAK inhibitors also have a significant side effect profile, including low platelets, immunosuppression, and an increased risk of skin cancers. It's obvious that we desperately need low-cost, low-risk treatments for MPN patients that address all the major issues in this disease. What if I told you that there's a low-cost, widely available, over-the-counter supplement called N-acetylcysteine that has strong preclinical evidence that it could be helpful for all three major clinical issues in MPN? In our lab, we recently found that NAC rescues a mouse model of MPN from deadly blood clots. Usually these mice die at about age 3 months from their blood clots. However, simply by adding N-acetylcysteine to their water, now some of these mice can live a full lifespan. A few years ago, we did an online survey of over 1,300 MPN patients, asking about their dietary habits, including supplements, as well as their symptom burden. In green, this means that the patient taking these supplements had a lower symptom burden. You can see that patients taking N-acetylcysteine had a lower symptom burden than those not taking N-acetylcysteine. N-acetylcysteine can be considered an amino acid, and as you see, people taking amino acids had a lower symptom burden than MPN patients not taking amino acids. NAC is a known potent antioxidant. This means that it can prevent the DNA damage that leads to leukemia. We are proposing a 22-week clinical trial of NAC in 50 patients with MPN. Our primary outcome is reduction in symptom burden. This is of key importance to MPN patients. Our secondary outcome is normalization of biomarkers of blood clots. There is a quick completion time for this study. We plan to enroll 50 patients by the end of 2021. Although this is a rare disease, over the past 7 years, I've developed a relationship with a cohort of MPN patients who is eager to participate in my studies. We plan to publish our results in early 2022. Because NAC is an over-the-counter, widely available supplement, this means that it could be widely adopted by MPN patients pretty much immediately after publication. We are able to execute this 50-patient clinical trial with only $50,000 because NAC is such a low cost and easily obtainable compound. Our team has the expertise that we need to complete this study in a short time frame. Me, I'm a physician scientist who has been committed to MPN for the past 15 years. As a physician scientist, I have the unique ability to translate my findings in the lab into clinical trials for patients that I care for in the clinic. For me, there is nothing lost in translation. At UC Irvine, we're very fortunate to have Susan O'Brien, a world expert in clinical cancer trials, leading our cancer clinical trial unit. And on the technical side, we have a very talented graduate student, Bree Craver, who's over the past few years developed and mastered sophisticated laboratory tests for assessing blood clotting in MPN patients and MPN models. This is the exact type of project that should be funded through Cures Within Reach. So critical because it may not have commercial value. The low cost and proven safety of NAC means it could be adopted immediately. This impact would be immense for MPN patients. The return on investment for each donated dollar of these patients is priceless. Moreover, NAC has the potential for a much wider use for cancer prevention, even in healthy people. And lastly, on behalf of University of California, Irvine and the Fleischman Lab, I'd like to thank you for listening to my talk, and I hope to get your vote.
Thank you, Angela. And you were mentioning the patients in your talk, and I think it'd be great if we heard from somebody who is a patient advocate. Dennis, would you like to ask the first question?
Sure, and congratulations, Dr. Fleischman, a very interesting presentation. I suppose when I look at it's a very promising study that you have presented, and from the patient's perspective, given the parameters and the way you've described it, it's imperative, I think, that it gets to patients as fast as possible. So my question really is, if we imagine the best possible circumstances as an outcome to your study, what level of any further research would be required, in your opinion, to get an approval for this indication? Or, as you've just said, with this publication in 2022, that itself could be sufficient?
Okay, so that's an excellent question, and I will try to answer it as best as I can. Because it's an over-the-counter supplement, it probably doesn't necessarily need an FDA approval as a drug. However, my goal would be at some point to have it incorporated into NCCN guidelines for patients with myeloproliferative neoplasms, such as something like a over-the-counter aspirin is currently on NCCN guidelines. For the outcomes of thrombosis or blood clots, which is our primary objective for treating polycythemia vera and essential thrombocythemia patients, those types of studies require thousands of patients with years of follow-up. So I think that from this study, obviously I'd like to do further studies with much larger cohorts of patients, but demonstrating that it has a significant impact on symptom burden in MPN patients, which is clearly a very important outcome among these people, is a good place to start, and can start the conversation with patients and get it more widely acknowledged by patients, and they can start to use it. Now, of course, there's always a sort of a downside in that if it's widely available, that there may not be necessarily an incentive for doing large randomized studies with the drug. So I do have to acknowledge that there's pros and cons for having something that's cheap and widely available and easy to get.
Thank you, Angela. Jeff, we haven't heard from you yet. Would you like to ask a quick question of Angela?
Sure. Angela, thank you again for the presentation. Can you just give us some insight on how you came up with the sample size for the proposed study? And what's the effect that you're looking for, active versus the placebo, that would give you a go versus a no-go?
Okay, actually, that's a great question. So we recently had a diet intervention study in 30 MPN patients, where we randomized them to either a Mediterranean diet or standard USDA diet, and then we followed their symptom burden. And so based on that cohort of patients and the impact of the Mediterranean diet on their symptom burden, we found that 50 patients is necessary to detect, with a 90% power, detect a 25% reduction in their MPN total symptom score. Is that what you were asking? So we're basing the power calculation on a recent dietary intervention study where we were also looking at symptom burden.
And assuming that that reduction was clinically meaningful in terms of they could really tell a difference in their symptoms.
So Angela, I can say yes. I mean, based on anecdotal emails saying that the diet had been life-changing and really improved. But I mean, that's to me, that seems that it caused an impact in their quality of life if they're, telling us that it's changed their life.
Excellent. All right, Angela, I think that's a great topic to continue talking about in the poster session afterwards. Thanks very much for your time. All right, next up we have Tysel Phillips, who is joining us today from the University of Michigan. We can have Tysel's presentation going, please.
Hi, my name is Dr. Tysel Phillips. I'm an assistant professor at the University of Michigan, and I'm here to talk about a study I'm very excited about and enthusiastic, and I think deserves your support: the repurposing of the base from mimetic venetoclax to treat newly diagnosed patients with mantle cell lymphoma. So to give you a bit of a background, mantle cell lymphoma is a rare non-Hodgkin's lymphoma, account for only 5 to 6%. The disease in itself is incurable, meaning the best we can hope was put the disease into remission at this current time. one of the biggest sort of needs in this area is to identify a treatment that is effective and be safely given to all patients afflicted with disease. Especially given that only 10% of patients will have what we call indolent disease, which tends to have the best outcomes and survival out of the other two subsets of this disease. Looking at the genetics, you'll see that cyclin D1 or the translocation 1114 is one of the first and key sort of pathognomonic signals in mantle cell lymphoma. But as you see following on the diagram, you'll see multiple other mutations that come along that will impact classic, blastoid, which is an aggressive variant, non-nodal leukemic, which is thought to be the indolent variant. And then you also have deletion of 17p, which can turn an indolent variant to aggressive. Treatment currently today is based on chemotherapy, which is basically drugs that are designed to damage DNA, flash and burn, napalm. They're going to kill anything in sight. So basically that counts for a lot of the toxicities we see with chemotherapy, as it kills anything that grows very fast. So in addition, with mantle cell lymphoma, we don't have a standard of care. So treatment is dichotomized between patients who are either young fit, who get high intensity treatment, versus those who are unfit. Now, one of the keys to this high intensity treatment is we think, and have shown this in several studies, that it induces what we call molecular remission, which puts patients into a deeper remission, which has been shown to have longer progression-free survival and or remissions. So one of the keys that we need to do to improve outcomes in patients with mantle cell lymphoma is incorporate treatments which improve the body's ability to eliminate the cancer cells by targeting specific targets or proteins and reducing the side effects. So the biggest issue we have is the lack of equality. Again, as I said, older unfit patients don't get the best treatment because they just can't tolerate it. So what we want to do is design a treatment regimen using some of these new molecular targets we have for mantle cell lymphoma to design a treatment that's more applicable to all patients, will induce deep remissions, and again, lead to the longest remissions. So given the strides we've made in understanding the biology of the tumor, certain medications have come upon that are certain targeted treatments such as lenalidomide. So this study in conjunction with rituximab has been studied and approved in patients with relapsed refractory mantle cell lymphoma, but has been studied in newly diagnosed patients and been shown to be an effective treatment with response rates above 80%. The downside to this is that the treatment takes a very long time to reduce remission, so patients are still suffering from the disease. The treatment is given indefinitely, and also they didn't look at molecular remissions, relying solely on radiographic remissions. So what we want to do is to improve some of these aspects. We want to speed up the time to remission, induce deeper remissions. We have the ability to stop actually the treatment and not treat patients indefinitely, and also lengthen the time patients can remain off the treatment. So to accomplish this, we want to add something to lenalidomide that doesn't sort of make the toxicity profile of the drug worse. So with this, we looked at the medication venetoclax, which is an oral BH3 mimetic. So basically how venetoclax functions, in the tumor cell, BCL2 sort of sequesters beta-bacs, which are pro-apoptotic proteins, and prevents them from causing cell death. So venetoclax, when it's ingested, sort of mimics beta-bacs and sort of binds to BCL2, which frees beta-bacs to cause cell death. So with this, again, we have a different mechanism of action, a different toxicity profile, and something that can be synergistic with lenalidomide. Ongoing clinical trial using venetoclax with lenalidomide rituximab is currently enrolled 18 to 28 patients. Today, we've seen great responses. We've seen acceleration of time to remission with remissions within 3 months, molecular remissions within 3 months. We're seeing very little toxicity with no dose-limiting toxicity noted, and all patients basically tolerating this regimen to the date. And we have response rates of over 90+% in the patients we've treated. So what we want to do is further enhance and sort of needle down to how effective this regimen can be. So in this situation, what we want to do is expand this study to a phase 2. But in order to do that, we need to do further things. So one, we do want to add additional time points to look for molecular remission. We also want to further investigate the genomics of the tumor DNA to figure out, if we can determine who is responding, why they're responding, and the difference between the early responders versus patients who responded later. So currently the trial is being supported by the University of Michigan Clinical Trials Unit. We have a grant from the World Cancer Center, and we also have funding from AbbVie. Additional funding from Cures will help us complete the enrollment of the clinical trial, add these additional time points to look for molecular remission, and perform this genomic test to look at home exome DNA of the tumor, again to help us identify the tumor cells, the factors of the tumor, which will help us to determine who's responding, why they're responding, and sort of speed of response. So due to these early favorable results, again, as I stated, we look to expand this trial and add another additional 47+ patients to formally compare to the Nordic regimen, which is our standard regimen of chemotherapy used in a multitude of institutions, which involves high-dose chemotherapy, inpatient and outpatient, autologous stem cell transplantation with 30 days in the hospital at least, and then maintenance rituximab. So the team itself consists of myself, Tyson Phillips, I'm a lymphoma specialist, Dr. Melody Kendarpa, who's our lab technician, who's also helping as far as a multitude of the preclinical work, which led to the development of this trial, and a multitude of tests as far as processing samples needed for the study. And Zanetta Nicolescu-Koleska, which is our BH3 expert, and who's performed the BH3 profiling that we looked to determine the sensitivity of the patient's treatment to venetoclax before starting treatment and while on treatment. So our expectations is this regimen will be designated as a valid off-label treatment for the National Comprehensive Cancer Network, which is a collection of comprehensive cancer networks in the United States, would put forth guidelines which are followed by insurance companies. We plan to complete enrollment of the phase 3 study within 2020, if not for the current pandemic, which has affected our clinical trial enrollment. We feel we would have done completing this trial by now. After completion of the phase 1B study, we plan to open and expand the phase 2 expansion, and we'll hope to complete that enrollment within the next 18 to 24 months, as we will add additional sites. Achieving the aims of this study with this additional expansion will allow us to substantially increase the number of patients able to truly receive effective frontline therapy, offer an improved safety profile compared to chemotherapy, eliminate the needs of these patients to have undergo high-dose chemo and autologous stem cell transplantation, reduce the amount of time patients need to spend in the hospital, and overall decrease the financial burden on our health system. With that, I'd like to thank you.
Excellent. Thank you, Tysel. That was great. Brian, you've been waiting so patiently for me to call on you. So why don't you take the first question here?
Thank you, Dr. Phillips, for that really important presentation and for submitting this proposal. Can you tell us a little bit more about your dosing rationale for venetoclax in this patient population, particularly related to the ramp-up approach that you've used?
Well, our ramp-up approach is a little bit different from what's currently in the investigator brochure, where they normally recommend 20, 50, 100, 200, and 400. When we started the trial, there wasn't really a specific ramp-up instruction for mantle cell lymphoma. So we assumed that it would respond similarly to what we've seen with CLL, but we didn't necessarily want to, or thought we needed to start the 20 milligram dose. So we did start the study with the 50 milligram dose. We didn't notice any issues, and when the formal guidance came out about starting at 20 milligrams, we had a discussion with AbbVie, and based on the number of patients we'd already treated, they were comfortable with us continuing with the 50 milligram dose. Now, the rationale for the dose ramp up is that specifically when you use these BH3 mimetics, you can sometimes cause massive tumor lysis, which when they looked at the original studies with CLL and they ramped up the dose too rapidly, they had deaths as patients weren't unable to clear the tumor breakdown products. So for safety reasons, you have to do a slow ramp up in most of these tumors who are very sensitive. So mantle cell lymphoma being one of the sensitive tumors, we do have to do a ramp up, just not as slow as they noted in CLL. And part of that is probably because even though there is mantle cell lymphoma noted in the circulating blood, it's just not of the quantity that you see in CLL, so that you don't get the massive shedding into the blood that you see with CLL patients.
Thank you.
Thank you. All right, Dennis, how about you take the next question?
Okay, yeah, great presentation, Dr. Phillips. My question, I suppose, bringing it back to the patient side of things, is really around quality of life, because given, say, the potentially relative positive side effect profile of venetoclax compared to the current standard of care, do you think that, or are you going to capture any quality of life data which may at a future date be able to help, you know, in the assessment of the clinical benefit of the treatment itself?
That's a very good point. We plan when we do the expansion to we'll have a much larger patient population to do a quality of life assessments, because again, the assumption is the fact that we are not seeing the nausea, the vomiting, the hair loss, the GI symptoms, and we're not having the rampant onset of infections that can sometimes come with chemotherapy, that at least the quality of life will be improved. And also we're keeping patients out of the hospital because they don't have to come in even for chemo treatments, which depending on the age of the patient, and part of the treatment regimens require inpatient stays and the avoidance of an autologous stem cell transplant. So we do plan, like I said, with the phase 2 to do a formal quality of life assessment, because we'll have about a collection of at least 70 some odd patients.
Okay, thanks.
And Maureen, if you have a really quick question, we have time for one short one.
Oh gosh, I don't know if it's really quick. Could you just maybe just very quickly address the time that this could impact patients?
Yeah, so just from what we've seen, the responses we've noted so far have been comparable to chemotherapy. That's not something we aim for. We assumed that we would half the response time that we saw with lenalidomide and rituximab alone, but we've actually even improved upon that. But we're seeing best response in some patients within 3 months and resolution of clinical symptoms within a couple of weeks. So that was even surprising for us.
Great. Well, thank you very much. That was a fantastic presentation. This is not going to be an easy decision for the attendees at all. So last but certainly not least, we have Kathleen Sakamoto from Stanford University. I'm giving her a talk. If we can get that video going.
I'm Kathy Sakamoto, and I'm a pediatric hematologist oncologist. I treat children with blood diseases and cancer. This study focuses on a repurposed drug known as niclosamide to treat pediatric acute myeloid leukemia. Pediatric AML is a form of acute leukemia, which is the most common type of childhood cancer. It represents about 25% of acute leukemia patients in children, and less than 60% of children with AML survive. There are very few treatment options for children who relapse, and less than 20% of them survive. The treatment for pediatric AML causes hair loss, weakened immune system, and many months in the hospital. Oftentimes, children cannot go to school, play with their friends, or do their normal activities because of their treatment or complications from their treatment. AML represents 700 cases or 8 million children with AML diagnosed per year. It is a rare orphan disease, and drug companies are not interested in developing new drugs for pediatric AML. If our approach is successful, we can use this globally for over 29,000 adult patients per year. The current treatment for pediatric AML is intensive chemotherapy, which are literally poisons and have many side effects in growing children and also late complications in survivors, which impacts their quality of life. The treatment is expensive, with intensive chemotherapy or stem cell transplantation costing somewhere between $500,000 to $1 million. Current therapy for relapsed AML have limited to no efficacy. And new therapies, such as immunotherapy, will not be available for at least 2 or 3 years for pediatric patients. The proposed treatment involves targeting a protein known as CREB. Our lab found that CREB is overproduced in AML cells, and this is associated with the worst prognosis. Therefore, we developed a small molecule to target CREB and inhibit its function in collaboration with a colleague at Oregon Health Sciences University. Unfortunately, this molecule is not ready for human use. So in collaboration with a medicinal chemist at Stanford, we identified an FDA-approved drug known as niclosamide, which, as you can see, has very similar structure to the small molecule CREB inhibitor. Niclosamide is an anti-tapeworm drug that's FDA approved and remains on the WHO's list of essential medications. It was withdrawn from the US market in 1996 due to low usage from better drugs for parasites. Interestingly, niclosamide has also been recently reported to potentially inhibit SARS-CoV-2 or coronavirus. Niclosamide is well tolerated in children, and it's not toxic, with the only reported toxicity being GI. Now, niclosamide in preclinical studies has been shown to inhibit cell growth in culture of AML cells, decreases CREB activity, and doesn't affect normal blood cell production. It importantly suppresses AML progression and increases survival of mice that take niclosamide every day in my mouth. Niclosamide also works together well with chemotherapy drugs to enhance killing by chemotherapy drugs such as cytarabine. Our study is a phase 1 clinical trial for relapsed refractory pediatric AML. The primary objective is to study safety and a recommended phase 2 dose of niclosamide in combination with the chemotherapy drug cytarabine in patients with advanced myeloid leukemia. The secondary objective is to evaluate the pharmacodynamic effect of niclosamide in myeloid leukemia cells with or without cytarabine. In other words, is the drug hitting our target CREB? And finally, the exploratory objective is to evaluate the pharmacokinetics of niclosamide in children with relapsed refractory AML. This is a clinical trial that involves dose escalation of oral niclosamide once or twice a day for 14 days, followed by 5 days of cytarabine. Patients who are over the age of 34, weight of 34 kilos and age of less than 25 years are eligible. The sample size is approximately 16 patients to evaluate 3 patients per dose level during a 2-year period. The budget for the entire clinical trial will be approximately $1 million. However, we have additional funding from the Leukemia and Lymphoma Society and Cure Childhood Cancer to support this clinical trial. However, we do need additional funding for pharmacokinetic and pharmacodynamic studies, data analysis, and salary for a biostatistician. The team at Stanford includes a clinical team, including myself, Dr. Norman Lacayo, who is Section Chief of Oncology and Director of the Poetic Consortium, Eileen Monge, who is our pediatric research nurse, and our research team includes Carrie Davis, who's Assistant Professor of Pediatrics, who will help us with the pharmacodynamic studies, Adam Frymore, who is a clinical pharmacologist, and Haley Headland, who is a bioinformatician who will perform the data analysis. The impact of our study is high because our clinical trial will potentially provide a non-toxic, effective treatment option for children with relapsed refractory AML and allow us to go to phase 2 trials. It could potentially improve the survival and quality of life in our patients since the drug can be taken at home. The treatment is inexpensive, $9 per pack, which is 4 tablets, compared to $500,000 or $1 million for intensive chemotherapy or transplantation. And if niclosamide proves to be well tolerated and effective, this would globally impact approximately 6,000 to 7,000 relapsed AML patients per year. So our clinical trial has the potential to change the lives of many children and adults with AML. Thank you for your attention, and I'm happy to answer questions.
All right, thank you very much, Kathy, for that presentation. Scott, why don't you start us off with the questions?
Yeah, thank you, Kathy. Really great presentation. The drug you're proposing to study, niclosamide, is, as you said, is an antihelminthic, and this class of drugs, they're by design, they remain in the gastrointestinal tract to kill worms as they pass through. And so in other words, as a class, there's very little of an oral dose of these agents that gets into the bloodstream to be able to work systemically. And I know there was a group that studied this drug in metastatic prostate cancer, and they showed very low plasma concentrations of the drug, and were unable to show a response in this patient population. Could you comment for the group in terms of, particularly around your preclinical studies, you know, the evidence you have and the confidence you have to be able to study this drug in these children?
That's an excellent question. In our preclinical models, which are patient-derived xenograft models where we inject pediatric AML samples, and we treated the mice on a daily basis for several weeks, and we clearly saw a decrease in circulating blast cells, including AML stem cells, and a decreased burden in the bone marrow. So, and these mice were given the drug by oral gavage. So I understand the other data that you're mentioning with metastatic prostate cancer. And I also think that our patient population is quite different in pediatric patients who receive chemotherapy. And, you know, we don't know really what the gut integrity is in these patients. So it's very possible that in our patient population, we may see a difference in absorption. And past literature has suggested that, in theory, the amount of drug that's absorbed is potentially anti-tumorigenic. So I still think it's worth performing this phase 1 trial, at least in our patient population, for those reasons.
All right, great, thank you.
Great. Thank you. And then Jeff, if you want to ask a final quick question of Kathy, we have a little bit of time for that.
Sure, let me just get it off of mute. So Kathy, what do you see as the key no-go from this study? What would you expect to see that would give you confidence in moving forward in a quantitative way?
Yeah, that's a great question also. I think that we're going to be monitoring the peripheral blood counts in the bone marrow of these patients, at least the peripheral blood counts weekly, bone marrow assessment, at 0 and 2 weeks and 4 weeks. And I think we'll be able to assess the degree of disease burden. If all the blasts do not disappear, or the patient has stable disease, is able to take the drug to control their disease at home, I think we've accomplished something for the patient and the family, in terms of the quality of life. But if we can also sustain a certain level of minimal residual disease, and these patients can go on to go to stem cell transplant or have another form of therapy that will hopefully become available for them.
Great. Thank you very much, Kathy. And I just want to give a virtual round of applause to all of our finalists for their hard work, not just today, but in leading up to the event. We had a lot of work with them, a lot of prep, so thank you. And also to our expert panelists for asking such fantastic questions, such thought-provoking questions. And we hope to see more of that going on in the poster session coming up again right after we finish here. So that concludes our pitch and Q&A section. Now we move on to the voting section, and to talk to us about that, I'm going to turn it back over to my colleague, Barbara. Barbara, are you there? Barbara, you're on mute.
It says I'm not.
We can hear you now.
We can hear you now.
Can you hear me?
Yep.
Okay, wonderful. Excellent. So thank you so much. And as Claire said, it was just an amazing, amazing job by everyone. When we selected these finalists, we knew it was going to be a rough choice for attendees to make, but it certainly has been a rough choice. Just listening, I'm still not sure who I'm going to vote for. In fact, they were coming along so well over the last month that we've been working with them that our team at Cures Within Reach has been hard at work trying to find funding for the second place and even possibly the third place winner today. We are in the process of hopefully securing a challenge grant. So if anyone in attendance today might be able to add to that challenge grant and get the second place and possibly the third place funding, please do reach out to Cures Within Reach, either Claire or myself, if you're interested in helping us fund the second or the third place winner today. With that, again, I encourage everyone to visit each of these 5 finalists in the chat rooms. If you are still not yet sure where your vote is going to go, that's exactly what the next 45 minutes are for, is for you to go into the chat rooms, ask the finalists some questions, and then help make your decision a little bit easier. From a process perspective, what's going to happen, before I tell you about that, let me not forget to thank our partners in this. We're extremely grateful for our funding partners. We're grateful for our industry partners, both Horizon and Recordati, as well as the partnership that we have with Global Genes, with the UPenn Orphan Disease Center, and this event as well. So again, I want to remind everyone you have 45 minutes to vote. The vote hyperlink will be listed on the screen in just a few minutes. If you keep your Zoom screen open, this will be listed. The next slide I'm going to show you is going to be listed for and open and available to you. We are also going to be putting this hyperlink into the chat screen shortly, so that you can always go back to the chat for the Zoom to find out how to vote. Each person has 1 vote only, so you can't vote more than once. The winner is going to be announced at 11:20 Eastern Time tomorrow, so please join us for that. As a reminder, as you've seen on the screen here, these are the voting considerations. And if everyone's ready, I'm imagining everyone's ready to vote now. the following way. You just go to any different internet platform, type in all lowercase, what you see here, bit.ly/CWRVOTE rare 20. So that's CWR vote rare 20. Again, the voting is now open for the next 45 minutes. And I do want to give another virtual huge round of applause for our finalists today. They did a spectacular, spectacular job doing this virtually. With that, I'm going to hand it back to Susan from Global Genes, who's going to make some closing remarks for us today.
I'm going to unmute. I think we're kind of in a pattern now. So thanks, Barbara, and thanks, Claire. Thank you to all the panelists and the finalists and the Cures team. Really, so much work went into this. Thank you so much. And thank you to our presenters both here and throughout the day for sharing their research and their perspectives and their insights. It's been such a wonderful first day at the Rare Drug Development Symposium, and on behalf of Global Genes and the Orphan Disease Center, we are truly appreciative to everyone for joining us today and sharing such great questions in the sessions and in the roundtables too.